Hello everyone. Today I am going to talk about biosafety levels. When we are talking about biosafety, it becomes very important not only in a microbiology lab, but it is effective in all the non-clinical labs. Here we are targeting the safety of personnel, organisms and the environment in a non-clinical or non-medical background setting. If you look at what is the meaning of biosafety levels, before we begin to talk about biosafety levels, let me just introduce a little bit regarding importance of biosafety. If you look at non-clinical laboratories, they are dealing with quite a lot of stuff, things like microorganisms, which include fungi, viruses, bacteria, everything. Now this becomes very important at one point of time, that how are we going to correlate the safety of the personnel as well as see to it that we are not contaminating the environment and no harmful organisms are escaping either into the environment or into any of the sewage systems, wherein it can potentially cause risk to the environment and all the people or all the stakeholders around. So when we are talking about the biosafety, they are always categorized in 4 different levels, with 1 beginning with the lowest level and 4 being the highest level of risk. Here we are talking about the risk that is involved. Low risk organisms that are involved in the research that is being done in the lab, and accordingly we go to the high risk organisms. If you look at this pyramid, look at the bottom of the pyramid, it is the largest, which is also telling that the number of labs throughout the globe, if you see, are maximum in the PSL1 level. Why? Because they include labs which are in educational institutions like schools, colleges, and also research institutes. But when it comes to research institutes, that is where actually the segregation starts to BSL2. When we are talking about BSL2 over there, we are talking about organisms which are involved having moderate risk. So there is a chance that there could be some risk involved, but it is not really very severe. Now when we are coming to BSL3, the number of laboratories are also reducing, and that is why the size of the trapezium that you see is slightly smaller, as well as the kind of regulations that are required are also more stringent. When we come to BSL4, that is the highest risk kind of a laboratory setup, wherein the organisms that are involved are high risk. That means they have a high fatality rate. The organisms which are involved can very easily spread through air, or which can, if spread or if they are contaminating the environment, they can cause really serious problems. And those type of labs are very few. That is why we are looking at a very small triangle at the top. Usually these are labs which are government labs or regulated entirely through the government or through such global agency like CDC or WHO or another such international organization. Now when we are talking about the different kinds of level, then each of the biosafety level will be having its own requirements. Basically, we are talking about 3 different ways. That is, looking at laboratory practices, safety equipment, and the facility construction. If you are looking at laboratory practices, that means what the things are normally done in a laboratory, what all things change slightly as and when the biosafety level changes. Similarly, when we are looking at the safety equipment, what are the different kinds of safety equipments that are used, including the laboratory instruments as well as the personal protective equipments that are being used by the personnel of the lab, and lastly but most importantly, the facility construction, the way the facility is constructed. Now if you look at the BSL 1 laboratory, which I told you, it is the most common kind of lab which we see in all the schools, colleges, or any such educational kind of a setting, or basic labs which are very common for, say, used in agriculture or many of the research institutes. Over here, the kind of organisms that are involved, or the kind of plant material or biomaterial that is being used is of low risk category. It will not usually cause a very serious harm, and if there's some kind of slight exposure, it can be very easily just cured with mild medication. So it is not going to be a serious kind of a trouble. And that is why we are having BSL 1. And over here, the kind of laboratory practices are very basic. That is, a simple clean table has to be used, and it should be free of clutter. The workbenches and floors and walls should be non-porous, easily cleanable. The segregation will be very simple, like for storage of chemicals there will be a segregated area, for instrument there will be a different area, for washing there will be a different area, basic kind of thing. And then the PPEs will be there's not very strict norms when it comes to PPEs. Basic PPEs like gloves and eye protection or safety goggles are usually enough. Only sometimes if there's some chance of aerosols, that is when the masks are being used, and sometimes masks are not used at all. And then basically when you talk about facility construction, that also is simple, just having a simple entry and an exit, and many times people freely go in and out, and they are just having a separate washing area kind of thing. That's it. Now when we come to BSL 2, over here there is slightly more stringent kind of you can say facilities that are being followed. What are all the things? First, when you talk about the kind of laboratory practices that are being done, if you look at that picture, in this picture you are seeing A, B, two kinds. Now what are this A, B? We'll see in the next slide. If you look at the laboratory practices, that is basically when work is being done at that time, there should be a facility to keep the lab closed. No one should just enter any time and as per their wish. When it comes to safety equipment, that is where I talked about A and B. Over here, they are not just using a face mask, they are using an entire shield that is covering their entire face. Okay, so there's a chance that yes, aerosols are produced. There can be splashes if they fall on the face or on hands, they can cause trouble. The kind of place where they are working is a biosafety cabinet. Nowadays, more well-known are laminar air flows, because they are the most common equipment of segregation nowadays and sterilization nowadays. And then when we are talking about facility construction, the doors should be self-closing. And over here, as I told you, if you look at the safety equipment, because of splashes and all, there's a risk that is directly related with a sink and an eye wash. So that should be readily available. Now when we come to BSL 3, please look at this picture again over here. Look at the safety equipment or the PPEs that this laboratory personnel are wearing, the kind of way in which they are working, the way in which the biosafety cabinet is very, very contained or closed. Now over here what happens is, these are the kind of organisms that are involved. They will be having a potential to cause disease, a potential to cause harm, and serious harm at that, requiring hospitalization or some serious kind of a malady, and sometimes having the potential to become very contagious and spread throughout the community wherever the exposure is there. And that is why we have BSL 3. Example is Mycobacterium tuberculosis, causing tuberculosis, as you all know. Now let us look at the different kind of laboratory practices. surveillance is required. When I am saying surveillance, that means they should be regularly given medical checkups, and if required, vaccination should also be given. Wherever possible, vaccination should be administered to the laboratory personnel. So safety of the laboratory personnel is very, very important. Stringent training to all the laboratory personnel, not only on the basis on say technical terms, but regarding safety practice has to be given on a regular basis compulsorily. Now we are talking about safety equipments. Over here, respirators may be worn, okay, and there should be a complete cover of the body from exposure. Okay, and that is why we can see disposable gloves. So you are not supposed to reuse them. You don't have say chance that okay, you just autoclave and reuse the kind of things that you've been using. Even the lab coat cannot be reused without appropriate autoclaving. Okay, and then when we are talking about facility construction, over here, besides the regular eye wash and sink that we had talked about, over here we are talking about a regulated environment wherein even the air circulation is regulated, so that there is not a potential of the microbes or the organisms escaping through exhaust or air from say the biosafety cabinet. So these should be self-locking doors. That is one more important thing. The entry and exit is highly regulated. Not anyone and everyone can enter. Only authorized personnel are allowed to enter, and that is how more stringent kind of facility is required, or more control is required, so that when who is coming and going, that can be recorded, and everything can be kept under check. Now we talk about the BSL-4, the smallest triangle, the least kind of laboratories that you can see all throughout the globe. There are very few BSL-4 level laboratories all throughout the globe. Say about 20 to 30 labs only are there throughout the globe which are classified as BSL-4. Why? Because over here, the kind of organisms that are involved, they have the potential to be converted very easily into biological weapons. For example, anthrax virus. It was once upon a time used in World War II as a biological weapon, and it created a havoc. So we don't want those kind of things happening. But research on these kind of viruses for medical use or for healthcare is very, very crucial or important. So it cannot be just removed altogether. And that is the reason why BSL-4 category is very few labs throughout the world. Before the start of the project itself, proper kind of regulation has to be followed. There have to be approvals on a very frequent basis. Auditing will be conducted to see to it that all the safety measures are being followed. Now look at the picture over here. Look at the suit that this lady is wearing. It's a complete body suit, and the thing that you can see from going from its inside to outside, there's a wire. Now it is a full body pressurized suit. Now since the entire body is covered right from top to bottom, it doesn't have breathing space, and that is the reason why the air is being circulated, and that is the reason why it is called body pressurized suit. The entire body is covered completely. Before entering the lab and at the exit of the lab, there is a shower cubicle. Over here we are looking at a full body air suit, wherein the complete coverage of the personnel is there in these kind of laboratories. Safety is the safety training is mandatory. It is very frequent, and it has to be done on a monthly basis. There is no excuse, or there cannot be any excuses for that. Now when we are talking about the facility construction, usually what happens is these kind of laboratories are constructed a little separately or isolated from other general kind of the environment, or you can say other general building wherein many people can come and go, so that the entry and exit of the people can be highly controlled. It can be seen that there is only authorized personnel entering or exiting. There will be complete control or regulation of the air that is being circulated. The exhausted air is also treated and then allowed to escape into the environment. Whatever the water or whatever things are being used are also autoclaved. Each and every small thing is autoclaved. Many times what happens is, in these kind of labs, there will be a built-in autoclave in the wall itself. So the contaminated equipments will be put from one end of the wall in the autoclave, and on the other side or on the outside only after sterilization the equipment can be removed. Such kind of facilities are many times constructed in these kind of BSL 4 level of laboratories, wherein highly risk kind of organisms are involved, because they can cause they can spread and cause havoc. And if you remember, there is a rumor regarding COVID-19 being spread from such kind of a facility only, and that is the reason why these kind of regulations, the identification of BSL levels, these all have become very important nowadays. Thank you, and please study well.